Okay, so this is going to be a review of Unit 1 of genetics, and how about we just jump right into it? So, the first thing we are talking about is the three main topics of genetics. So, we have transmission genetics, which is basically studying how traits are inherited, and that's where you get like Mendelian and your Punnett squares. That's kind of like what does your parent pass to your child, that sort of thing. So that's transmission genetics. We have molecular genetics, which is kind of like what we're learning now, exactly what's happening in the DNA and the RNA interactions between DNA, RNA, different proteins, different enzymes, all sorts of that. And then we have population, which is looking at the population as a whole and kind of seeing how does what is the genetic diversity of a population, and how that can change depending on certain events, and kind of just how um, people evolve, um, or not people, organisms evolve. Another big thing to know is prokaryotes versus eukaryotes. So, prokaryotes, they um, do not have a nucleus, is like the main thing you were probably told, but they're just more simple in general. All of their DNA is free floating. So the DNA is basically free floating around there. And it's not arranged in those linear chromosomes that you usually see. That is not a eukaryote thing. No, that is not a prokaryote thing. Um, Eukaryotes have these linear um, chromosomes, but have yes, prokaryotes have circular chromosomes. Just remember that. That's the main difference we need to know for this unit is prokaryotes have circular chromosomes, eukaryotes have linear chromosomes, and prokaryotes are a lot simpler than eukaryotes. Eukaryotes have those membrane-bound organelles and a nucleus, and prokaryotes do not have that. Um, so viruses and cellular genetic systems is non-cellular genetic systems. So that's just some words you should probably know, but that viruses are not technically living by various definitions. It's still up for debate, but they need a host cell to replicate, is what you know. So viruses need a host cell to replicate their DNA or RNA, but they need a host cell to replicate their genetic material. And then like there's some non-cellular genetic systems. So that's talking about like horizontal gene transfer. So that's not when parent to child is the regular gene transfer. That would be like vertical gene transfer because it goes parent down to child. But what if you're transferring a gene from the mitochondria to the nucleus of a cell? Um, that's not hereditary. That's not like parent to child. It's just like one place in the cell to another place in the cell. That's kind of what they're talking about there. So, I'm gonna clear some of this off. So genetic model systems. So these are organisms that we use to study the effects of genetics and that we can manipulate easily. And some common ones are rats. Drosophila is a fruit fly, basically. There's yeast, E. coli, mouse, zebrafish, and the reason why we pick these is mostly because of how easy they are to breed, how easy they are to handle. Um, Humans take a long time to reproduce, but Drosophila can reproduce in a matter of days. So that's really good for seeing like gene changes from each generation. Um, we like to use rats because they mimic general mammals. So a lot of testing for stuff that goes on in mammals, say like vaccines and drugs, they test them on mice because they're a good model organism of the animal. We share a large amount of DNA with mice. Yeast is for general eukaryotic cells. They're a very basic eukaryotic cell. And so that makes them ideal because there's less variables going around with when you want to test something. So you could just test it with yeast. Zebrafish, good species. especially if you're looking for something that has a vertebrae, but still keep it a simple species. E. coli, a really great general cell prokaryote to test on. And yeah, we already said this, but it's a big factor of it's easy to take care of them. They reproduce quickly, so they're ideal study units, basically. Okay, so moving on. Basic unit of inheritance is a gene, basically. And what are alleles? So, a gene can have a gene is, say, this codes to a protein to an enzyme like insulin. And so alleles are basically the variations that the gene can have. So if the enzyme is insulin, an allele could be it produces insulin, or the second allele could be it does not produce insulin. Um, Another example of this is in we could just use like snakes, for example. They're one of my favorite examples. We could have a gene for albinism, right? That it's the gene that controls the amount of melanin in skin. And one allele could be normal amount of melanin. So that would be like that would lead to like a darker colored snake, like black or brown. And then another allele could be the lack of melanin. So that would turn into like a yellow snake, and that would be albinism. So basically, an allele is a different variations of one gene, if that makes sense. So phenotype versus genotype. So a phenotype is basically what you can see and observe. So that would be like brown hair, green eyes, um, purple flowers versus pink flowers, just something that's observable, and it's more of a quality rather than a quantity. It can be a quantity, but when we think about it, it's more like what are observable traits you could see versus genotype is what the actual genetics are. So, like, if we had like a Punnett square, like our genotype would be like, oh, are we capital H, lowercase h, or are we capital H, capital H? So that's just the genotype is telling us what alleles we have, whereas the phenotype is like, oh, do you have brown hair or do you have blonde hair? That's kind of the split between phenotype and genotype. Um, so what are genes made of and where are they located? So genes are made out of nucleic acids, um, which are your subunits of like DNA and RNA. That's your 5 carbon sugar with your phosphate and your um, base, um, like would be ATGC. So that's a nucleic acid. That's what DNA, RNA genes are made of, and they are usually located in prokaryotes. They are located just in the cell. In eukaryotes, they're either located. DNA is located in the nucleus. RNA you will find everywhere. And there's also some DNA in mitochondria and chloroplasts. Um, so yeah. Okay, so this is the portion of an overview of the structure of DNA and also the history. So first of all, an important name to know is Chargaff. Chargaff was the guy who found out that A pairs with T and G pairs with C. So, that base pairs were in pairs, they weren't just random, and that they form some kind of code. And that plus T and G plus C are not in a fixed ratio. So in a mouse, you could have maybe 50% A plus T and 50% G plus C. But then in a dog, you could have 30% A plus T, 70% G plus C. And so each organism is different and they do not have the same amount of A and T or G and C. And A and G are purines, C and T are pyrimidines. So purines kind of look like this. So they have one like structure and then they have another structure next to it. And then pyrimidines are just one structure. So how I remember this is that purines stay pure because they have extra defense. They have this little guy over here that defends them from all of the bad stuff. So they stay pure. Um, Now, and A and G are purines. Um, and like purines are like better, and A is the first letter of the alphabet. I know that's a bit of a stretch, but that's kind of how I remember that. Okay, next we have Griffith. He basically did an experiment that had bacteria, that was virulent bacteria, pathogenic bacteria, so bacteria that made the animal sick, and bacteria that didn't make the animal sick, and saw that when that the and he saw that the pathogenic bacteria would give genetic material to the non-pathogenic bacteria, make them pathogenic. So the bacteria that would not cause sickness would get genetic material from the bacteria that did cause sickness and become bacteria that also caused sickness. So that is a phenomenon known as transformation. And he also found out that DNA is pretty heat stable. Avery, McCarty, and McLeod, they treated strains of bacteria with different enzymes that destroyed either protein, RNA, or the DNA. And whichever group did not transform the surrounding bacteria, this principle of transformation we learned about in the previous experiment, if it did not transform the surrounding bacteria, then that means that they had killed the material that contained genetic information. Um, so, yeah, and then we have Hershey and Chase, which confirmed that DNA is a genetic material responsible for transformation in hereditary information. Now, this is the important takeaway. Takeaway from basically these three, this is the most important. Out of Griffith, Avery, McMarty, and McLeod, Hershey, and Chase, the most important thing is that DNA contains phosphorus, but not sulfur. And sulfur is associated with proteins, and proteins also do not contain phosphorus. So basically, um, DNA has phosphorus, proteins have sulfur. They do not have any overlap with each other. That is a good way that is a way they distinguished proteins and DNA, and something you should remember. It's a big takeaway. Um, If you want to look more into their experiment, you can look it up. But this is the most important thing, and I don't want to make this video super long. So then we also have DNA structure. This was discovered by Rosalind Franklin, who took the image, and then Watson and Crick, who actually put the structure together based on that image. And a big thing about DNA is it's anti-parallel. That means that it's not two strands in the same direction going together, just parallel. It's actually one strand in one direction, so this is an X out, and one strand in the other direction. Basically, one's going one direction, the other one's going the other direction, but they're still parallel. That's what that means. Hydrogens between base pairs, so if we have, say, a purine of adenosine, then it would have two bonds with thymine. And those bonds would be hydrogen bonds. Now, importantly, A and T have two bonds between them, and G and C have three bonds between them. This means that G and C take more energy to break apart because they have more bonds. More bonds equals more energy. There's also different types of DNA structures. So the double helix we all know and love is called the B DNA. It twists right. It's spaced in a certain way. We know that that's a typical formation, but there's also A DNA, which is a really tight twist, and there's also Z DNA, which is a really loose twist, but it's also twisted the opposite way. This kind of arises from differences in DNA methylation, and it can lead to triple DNA. So just know that bDNA is not the only way that you will see it. It's not always double helix. Sometimes it can be a triple helix, which is kind of wacky and weird. We have RNA structure. It is single-stranded, but can pair on itself, and that's where we create these hairpin loops that are really important in material we'll discuss later. But they can like create all sorts of bubbles because they're just pairing with themselves. So say if there was like an A here and it saw a U on the other side, because RNA uses uracil instead of thymine, U instead of T. Important, remember that. And these were like, oh, I see each other. Let's pair up. So that's kind of what happens. Um, and this unusual pairing actually helps stabilize structure and sometimes gives the RNA functions, like in the case of this hairpin loop that we'll get into later. And also, some viruses contain RNA as their only form of genetic information, not DNA. Alrighty, let's now move on to central dogma, which is honestly the bread and butter of this unit because what we did before was kind of an overview, and now this is the really important stuff because it's the process of turning DNA into protein, which is basically taking that genetic material and taking it into something, making it into something that the body can use for various amounts of um, various amounts of things. We will go over them later. So first of all, before we get into that, we need to go over replication of DNA, because that's how we can continue to have our DNA. That's how we can, cells can divide. Um, so this is like how maintaining DNA. So replication is semi-conservative. This is important because that means that the template strand stays the same. Strand is the same. Uh, what was that? Same. And then, but then a new strand is created. So, it's semi-conservative. So, the new DNA will be half old DNA and half new DNA. There is complementary base pairing. That is basically the rules of egg pairs with T and G pairs with C. Um, and then we have three phases, which is initiation, elongation, and termination. We will get into these in the next few minutes. So, there are a few types of replication that we covered in this course. The first that we're going to talk about is theta replication. So theta replication is when you are making one copy of a chromosome, and only happens in bacteria, and it's with circular chromosomes. But the end result, you really just need to know what the end result is, which is two circular chromosomes, just two. You are only replicating and making one copy. That is what theta replication is. It only happens in bacteria with circular chromosomes. Um, that's pretty much it. I can't draw. Okay, so then we have rolling circle replication, and these are for like viruses and plasmids. So these are for what's it called? Uh, Pieces of genetic material that are much smaller than chromosomes typically. So I'm gonna try to draw this. It might go badly. There's always a better picture on the slides. So the main thing that you need to know is that this one creates lots of copies. Theta replication only creates one copy. This one creates lots of copies. Um, yeah. All right. So then we have linear, which is the one that eukaryotes use. And that is when we have linear piece of DNA, linear piece of DNA, bunch of replication bubbles will form on it, and they just get bigger and bigger until they merge together into two strands of DNA. Yeah, this kind of sums it up. There's multiple origin sites where the replication bubbles start. These things are called bubbles, and they just go. So it's like think of it as if you unzip a zipper, but from like multiple spots in the zipper. That's kind of what happens in linear, and that produces just one copy, just like the theta replication. So yeah, what are the actors involved in this? DNA polymerase, a big one. It synthesizes from the 5' to 3' side. The 5' side is where the phosphate is. And then the 3' side is where the hydroxyl group is. Um, but really you need to know, where does it go? 5' to 3', always, and a good way to remember this is it reads up, writes down. So it will synthesize DNA or write DNA from 5' to 3', so down in numbers, and then it will proofread from 3' to 5'. So it reads, so it's read up, write down. That's a good way to remember it. Yeah, and just remember this if you're ever given a picture. But yeah, this process is semi-discontinuous. This is a big word, but it's like really important. That semi-discontinuous and semi-conservative are the two words that you should probably know for the test because I promise you they will probably be on there. So this is talking about leading versus lagging. So do you remember again? We said that it writes from the 5' to 3', but it's a bubble. So if it was going, this is 5' to 3', it would go, because if it has to synthesize in this direction, but the way to synthesize is this direction, so it will go synthesize, and then it will go back to the start and synthesize, and then go back to the start and synthesize. So it's just kind of like synthesizing backwards. And then these are eventually linked together by something called DNA ligase, which we're almost about to learn about. So that's cool. So replication in bacteria, basically initiation, the origin site bound by initiator proteins, unwind DNA, provide a binding site for replication proteins like DNA polymerase. Yeah, that's pretty much what it says. Basically, there's proteins that latch on to the origin site, and then DNA polymerase can bind onto those initiator proteins, and then it's able to synthesize. Without the initiator proteins, the DNA polymerase would not be able to bind onto the DNA, and so it wouldn't replicate. So that's why the initiator proteins are needed. Okay. Replication components. This is a big one. I said in this thing, highlighted are ones are must knows. That means they have a high likelihood of being on the test. The other ones do not have a high likelihood of being on the test. I suggest you should probably know them, but if you're in a pinch, you should probably know the ones that I highlighted. Helicase unwinds, unzips DNA. I always think of this like helicase, helio, sun, super hot, super fast, burns through things, unzips, unwinds. I know it's a stretch, but it makes sense in my head. Hopefully that helps you too. Okay, so these are single-stranded binding protein DNA binding proteins. These prevent the DNA from zipping back together. Honestly, you should probably know this one. I don't know why I didn't highlight it. But yeah. There's topoisomerase. Top whatever. You can see it. I don't know how to pronounce it. But basically like the DNA is wound really tightly together, right? And if you ever have a rope and twist it really tightly together, it just wants to coil back in on itself. And topoisomerase, that's how you say it. Topoisomerase makes it so that it won't coil back on itself, and also the force of trying to coil back on itself will not break the DNA. So then we have primase. This is highlighted. You should know it. Um, basically, DNA polymerase is a little bit dumb, and it cannot synthesize. It doesn't know what's next. It literally just says, "Hey, I don't know what that pairs with." So RNA primers have to go in and say A pairs with T, and then DNA polymerase will be like, "Oh, yeah, A does pair with T. I'll put a T there." So basically, the primase kind of, like, sketches it out for the DNA polymerase, and then the DNA polymerase can do its job. So that's kind of what primase does. It just puts in a few RNA segments ahead of the DNA to just, like, show the DNA what it should be putting in there and what it should do. DNA polymerase 3. This is making sure there's enough energy to add the new nucleotides to the new DNA strand. New nucleotides being A, T, G, or C. There's also DNA polymerase 1 that removes RNA primers, qRNA, I know what to do now, and then replaces them with DNA. DNA ligase. Do you remember those Okazaki fragments we were talking about earlier? That basically combines them so they're all a smooth line instead of like a bunch of fragments. Clamp, it's kind of obvious to the name, but it holds DNA polymerase, that's full for short, to the template, and it also increases the rate of synthesis. And there's a clamp loader that holds like everything together. So the clamp is like a small thing, clamp loader is like a huge clamp. So yeah, that's kind of what you need to know. And about all of the enzymes that are at work, those are the ones you should know. In eukaryotic cells for replication, basically just like it's basically like what happens in prokaryotes, but like 10 times bigger. More than that, a lot bigger. Just it's basically the same, just like a lot more of helicase, a lot more of DNA polymerase because it's a bigger cell. So yeah, there's a problem with linear chromosomes, which is kind of concerning given that we have linear chromosomes. But the primer on linear chromosomes is never be able to be removed at the end. Um, just because of how it's replicated. And this means that we are essentially losing little chunks, like, I don't know, like 5 base pairs. We're losing little chunks of our DNA every time our chromosomes replicate, which is a big problem, right? Because what if that like last snippet of DNA contained something really important? Like you kind of want it to stay there, not just be like lost. But that's kind of what's happened. And so in order to fix that, we have telomeres. Telomeres are basically a bunch of repeats at the end of um, a DNA sequence. So it would just be like A, C, C, A, C. So it just keeps repeating and repeating and repeating. So that builds in some buffer. That doesn't really do anything. It just builds in buffer um, so that when we replicate, we're not losing useful information. We're just losing old information. This is something that is associated with aging. So obviously, as you age, your telomeres get shorter, and that's why we have telomerase, which tries to fix some of that problem. So it will add more base pairs onto the end of this um, in order to help the telomeres not shrink at a terrible rate. But so telomerase is elongates the telomeres. I know that's a lot of information. What you need to take away from the telomeres thing is linear chromosomes will lose base pairs every replication. So we have buffer built in called telomeres, and telomerase makes that buffer even longer, or tries to keep that from getting super short super quickly. One more thing before we hop into transcription, we have homologous recombination, which is basically when you're trying to, everybody has two pairs. We have 23 chromosomes, and each of those chromosomes had a pair. Say we're replicating these pairs at the same time. If a strand, while replicating, if a strand breaks, and the other strand breaks at the same time, then there's a chance that this strand actually starts replicating on this side, and this strand actually starts replicating on this side. So that's basically what that means. So there's lots of different ways this can happen. Uh, this is kind of like the basic example of it replicating of it's crossing over, but it can also cross over like 3 times. It can cross over like um 2 times. It's like a whole thing. But basically, all you need to know for now is it leads to genetic diversity, and it's really important in reproduction. So yeah, and then it stems from double strand breaks or single strand breaks. Um, it stems from breaks in the DNA during replication. Yeah, and that is our replication overview. Okay, so now we are going to talk about transcription. So first of all, here are some key players in transcription and translation that are all versions of RNA that we need to know. I had said, oh, these are the only ones you need to know for this unit. I was wrong. We cover more of them in other unit in later in this unit. So we will be going over those shortly. So first of all, we have mRNA. mRNA is probably the most common form of mRNA and something you probably heard before, but it stands for messenger RNA, and it basically is the go-between in gene expression, meaning that it is it comes straight off the DNA and goes to the ribosome. So it carries the instructions to make proteins. We have rRNA, and it's a type of RNA that makes up part of the ribosome. So a ribosome is composed of protein and rRNA. That's how that works. And then we have tRNA, which is also used in a ribosome, and it's basically like a key that matches the RNA to the amino acid. So it's like a match finder. Um, so here's something that you should probably know. Um, messenger RNA is complementary to the template strand and matches the coding strand. So if there's a question on the test about coding strands, it says something about coding strands, template strand, and the RNA strand. So the coding strand, so we have the template strand of DNA here, and the coding strand is up here, right? RNA is complementary to the template strand, which means that if the template strand says like ACG, the RNA will be UGC. U because RNA has uracil instead of thymine. So basically, RNA is complementary to the template strand, but it is the same as the coding strand. The same as in the same code, but all of the T's in the coding strand are replaced with U's for RNA. So that's what that means. Make sure you do not get the coding strand and the template strand mixed up. Think of coding strand as the actual code that the mRNA is, and the template strand is what the mRNA uses to create that code. And each strand can actually encode different genes. And how does this work? It's with alternative splicing, which we will get to later, but just to have that connection in the back of your mind for now. So, what are genes made of? I didn't mean to scratch that out. What are genes made of? We have a promoter, a coding region, and a terminator. So, we usually have, say this is the coding region, we'll have a promoter up here and a terminator down here. So that's kind of the entire gene. Upstream versus downstream. Upstream is this way, downstream is this way. That's what you need to know. We have RNA polymerase. RNA polymerase works similar to DNA polymerase. It reads up and writes down. So it reads from 3' to 5', synthesizes or writes from 5' to 3'. It has the capacity to unwind and rewind DNA. So in replication, we need all of these kinds of other enzymes like helicase, topoisomerase, all of these to keep it unwind and unzipped. And RNA polymerase can do this all by itself, which is pretty cool. It also does not need a primer. It knows what it's doing, and it has weak proofreading ability. So this is kind of like an all-in-one enzyme to do everything it needs to do to create that messenger RNA. Um, transcription in bacteria. This this is slightly different from eukaryotes, and we're going to go over exactly how it is. So, RNA polymerase binds to a specific sequence of the promoter. So that is usually the 10, -10, -35 boxes. If we're looking in the upstream and downstream, Negative 35 would be like down here, and negative 10 would be like here, and then the start would be like here. So that's kind of how those are located. And one thing that's really important about bacterial transcription is the Shine-Dalgarno sequence. That is unique to bacteria. It's not in eukaryotes. So if you see a Shine-Dalgarno, we're instantly thinking about bacteria and prokaryotic cells, not eukaryotic cells. So basically, there's also a thing called a sigma factor. This guides the RNA polymerase to bind to the promoter. Um, and we can start unwinding the DNA, and then our RNA polymerase starts going off and synthesizing all of that messenger RNA. So then we have termination. Now, 50% of bacteria uses row dependent and 50% uses row independent. Row dependent, row is a protein that basically separates the RNA from the DNA in transcription bubble. If the RNA here, the DNA here, it just cuts it off and lets the RNA float away while the DNA is now like this again. However, if we are then the road independent, then the mRNA actually forms a sequence where it kind of starts to attach to itself, and the hairpin is formed, and this hairpin structure is able to dissociate or remove the RNA from the DNA so the DNA can go back to being its normal state. All right, and then we go to eukaryotic transcription. This is pretty similar to prokaryotic. A main difference is it has no Shine-Dalgarno sequence and instead has a Tata box as core promoter. Tata box literally means it has nucleotides T, A, T, A, but we call it Tata because that's what it spells. This is necessary, basically, but it's also not sufficient. So we have a bunch of different transcription factors, which are a bunch of different proteins that bind to the Tata box and the promoter in general, and allow the RNA polymerase to bind and start working. So transcription cannot begin until full assembly of transcription factors. So basically, it's just a little more complex. It's not like one protein; it's a bunch of proteins. So yeah, we have 9 different proteins plus this protein. We have a bunch of other ones but that's not really that important. What the one you should know is probably the TBP, which is the Tata box binding protein, and it opens the DNA and allows RNA polymerase to start synthesizing. Elongation and termination, same as prokaryotes. Termination is not row dependent, so it is not dependent on a protein. Um, Basically, transcription continues, or basically, it's like how the hairpin is formed and then it breaks off, and transcription actually continues, but the resulting RNA is ultimately destroyed, so it's not used. We have different types of RNA polymerase, just like we have different types of DNA polymerase. So RNA polymerase 2 transcribes protein encoding genes in regulatory RNAs. So each RNA polymerase transcribes a different thing. It doesn't have a different function, it just is used in different areas of the cell. So this is protein coding genes and regulatory RNAs. This is the main one when we're talking about central dogma. RNA polymerase 1 transcribes rRNA genes. So polymerase 1 helps create ribosomes. So wherever ribosomes are created, basically, wherever ribosomes are made, RNA polymerase 1 is there to make rRNA. And then we have RNA polymerase 3, which transcribes tRNA. So basically, again, this is part of ribosomes. This is a part of the function of ribosomes, but it will make tRNA. Mitochondria and chloroplasts have their own kind of RNA polymerase because they have to do their own transcription, but it's basically the same. It's just like a mini cell. This is not very important. Summary. This transcription is the process of DNA to RNA. Promoters are important, and RNA polymerase is important. That's the broad overview of what you need to know for transcription. All right, some other things. It was initially thought that the transcribed RNA immediately just got translated to proteins. This is not true because we have something called mRNA processing. So we have introns versus extrons. extrons. So basically, when mRNA comes out, it's a whole long strand, right? But some of this is not useful. It's non-coding DNA. That means we don't want it to be in our instructions. So these are called introns, and we splice them out. So say we have an intron here, and an intron here, we splice those out. So then so if this was the blue, this was blue, this was blue, and those two are orange, then we splice them out and combine them to make one blue strand. And these are all exons, and they will go and code for a protein. And these actually use small nuclear RNAs or snRNAs to splice out the introns. So this is another type of RNA, and it is useful for splicing and mRNA processing. And there are a bunch of different types of them, but that's not really important. The important thing is that they exist and that they splice mRNA. Yep, that's what I just said. So alternative splicing is when you can splice introns and exons in different ways to make different genes or different proteins. So one gene can code for a ton of different proteins because you splice it differently. So if we went back to this example where we had blue and then we had some orange and then we had blue and then more orange. So these are the introns. They absolutely do not code for anything. What if we spliced out some of the blue here? Then, let's make this green then. So then this new green piece gets produced, and that's different from the blue piece. So that's kind of alternative splicing. And then this green piece will create a different protein than the blue piece because it has different code in it than the blue piece. That's kind of what the gist of alternative splicing. And the main takeaway of this is one gene can code for several different proteins. Mature mRNAs are when the splicing has already occurred, now they are mature, are also given poly-A tail, so they're given a bunch of A nucleotides, and a methyl GCAP to prevent degradation. I would say just remember the words poly-A tail, methyl GCAP, these prevent degradation. They also contain UTRs, which stands for untranslated regions. So the one at the 5' end, UTR, is for mRNA export translation. The 3' UTR is for stability and translation. So just know that UTRs are important for stability, translation, and like export. In eukaryotes, only mature mRNAs leave the nucleus, but in prokaryotes, processing does not happen. So basically, all that I just talked about does not occur in prokaryotes. Um, tRNA can also be processed, but importantly, main important processing that you need to know is addition of a CCA to the 3' end after transcription. This will allow an amino acid to bind to the tRNA. So that's the really important part. And you sometimes generate unusual bases, which we'll get into later. And the anticodon is complementary to the mRNA codon, which allows the tRNA to match an amino acid to the codon. So if the RNA said UAC, the anticodon would be AUG. And that's kind of how that goes. rRNA, all you need to know is part of the ribosome, and they can also be processed. Small RNAs, basically, they kind of go around and clean up the genome and help destroy unneeded RNA. Do you remember when we were talking about degradation, ultimately destroyed? That's what these small RNAs, siRNA and miRNA, help with. And they are also connected to CRISPR, but you basically need to know that they run around destroying things and they help clean up the genome. Alrighty, so now we're going to go into translation. So this was discovered by Beadle and Tatum. Just a fun fact, not something you really need to know. One big thing about this is the code is redundant. So if you see, I don't have one on hand, but I will put one up now. If you look at this chart, you can see that multiple codons code for the same amino acid. This is really important because then if the tRNA accidentally binds with the wrong codon, which is called wobble pairing, something you learned about in your breakout session, then um, it can still bring the correct amino acid to the polypeptide chain, even though it's slightly different. So the tRNA can have a different third nucleotide in its anticodon than the third nucleotide in the codon, and it will still be able to bring the same amino acid to the polypeptide chain. This is really important because it helps decrease errors and mutations have less negative effects. But yeah, anyways, that's why the redundancy is super important. It builds in protection against mutations and it enables wobble pairing to happen and also not create a bunch of problems for the organism. The start codon is AUG, and that's really important because that signals to the tRNA, okay, let's go, let's create a polypeptide chain and string a bunch of amino acids on. You should always know that AUG is the start of your codon sequence. There is a universal code. For the most part, there is no, this is the same across all organisms. There are very few exceptions. There are always exceptions in biology, but this one has a lot less exceptions than most. Okay, so tRNA. tRNA is a little thing. It's like a clover that looks like this. Very badly drawn, as per usual. But right here is where the codon is. It will be three little three base pairs. And then here we have CAA. This CAA is really important. It binds to an amino acid and adds it to the peptide chain. So this is where the amino acid goes. This is where the codon goes. So it's kind of like a matcher piece. Each tRNA carries a different acid. So we have a bunch of different kind of tRNAs to carry different amino acids. And how does the amino acid attach to the CAA end? It is charged. We call it it's charged with an amino acid. And this requires ATP. ATP is energy, adenosine triphosphate. So tRNA is charged with amino acid by enzymes, and these enzymes only recognize one amino acid. So this is again another way so nothing gets mixed up. Everything is compartmentalized to each amino acid, each codon. Nothing is getting mixed around. But yes, if the codon was CUG, it would be GAC. There we go. Now we're going to talk about wobble pairing, which I referenced earlier. Sometimes the tRNA can pair with codons that are not exactly aligned with the anticodon on the third base pair. So yeah, this again goes back to the redundancy in the code. But yeah, AGG could pair with UUU instead of UCC, and so therefore one tRNA can recognize several codons. And if this happens, it could still spit out the same amino acid, which is really important. Um, Same amino acid. Yes, so that is tRNA overview. Um, then we have the ribosome, which is made out of protein and rRNA. And it basically reads mRNA, then aligns with tRNA, and it catalyzes the bonds between the amino acids. I just read off the notepad. Basically, it will read the mRNA and then it will be like, okay, see, this is the ribosome, this is the mRNA. Then it says, okay, tRNA, come right over here. And then it will help polypeptide bonds form so that the amino acid chain can form. And that's pretty much its job to create proteins. Um, if you need a refresher on all of this terminology, an amino acid is a subunit of a protein. It is just the base unit of a protein. Peptides are the same thing. Polypeptide is when you have a bunch of amino acids linked together. And a polypeptide is what folds into a protein. So proteins are made out of peptides or amino acids, which join together to create a polypeptide, which folds itself to become a protein. Enzymes are a type of proteins. That's something that will help you to know is that enzymes are a type of proteins. Um, the code. Okay, so yeah, this is non-overlapping. and the frame is set by AUG. So if it was said like, I don't know, UAAC AUG, the frame does not start here at the first three or here, it starts here. This AUG sets the frame for everything else. Nothing happens until you hit that AUG and then it's go time. It also non-overlapping, so you will never have one that you go, oh, UAA, and then you go ACA. That's not how it works. It will always just be three at a time, and you will never overlap the three. Initiation in bacteria, the initiation factor will bind to that almighty Shine-Dalgarno sequence again. Initiation in eukaryotic cells, there's no Shine-Dalgarno sequence, and we use a start codon. And that's also part of a Kozak sequence. Not super important, you just remember the start codon. Formation, elongation, and termination. There are proteins that are called elongation factors, and they help with this. And release factors bind to stop codons, and that releases the polypeptide and helps the ribosome just kind of like fall off of that chain. What about proteins? Proteins are used in enzymes, transport, signaling, storage. They are super important. They are why we are alive. They are essential for literally everything. What a huge thing. Function of protein depends on structure. This is huge. I cannot emphasize how much this is used, but remember that. Function of the protein depends on the structure. If the structure changes, the function changes. And these proteins also self-fold into their final form by different interactions of the amino acids, because some of them will make hydrogen bonds with each other, or they'll make covalent bonds with each other, and they'll just bond together into and fold the polypeptide chain into its final form. They also have 4 levels of structure, so they're pretty complex. And basically, this all culminates into, in order for proteins to be made, and you can see a phenotypic difference. For example, if in order for the protein of melanin to be made to make your skin darker, genes for melanin must still go through the transcription and translation process of central dogma to create the proteins to show gene expression. This is video game actually that helps you learn how to fold proteins, and it's called Fold It, and you can see how there are a bunch of interactions within the protein itself, and that's why structure is so important to function. And now we're going to do a quick practice question. So what's said here is not very important. It's kind of how I'm going to walk you through my process of how I use the information I know and connect it to the question, which I think is the most important strategy to know, barring just knowing the material, is knowing strategies for how to link that material and apply it to the problem. So, this practice problem. A mutation in a cell causes RNA polymerase 1 to fold incorrectly, changing its structure. What effects would this likely cause? So let's look at the answer choices. We have proteins would no longer be produced. Nothing would happen because RNA polymerase's function is independent of its structure. tRNA would not be processed incorrectly. Ribosomes would no longer be produced. So we need to look back and say, what does RNA polymerase 1 do? This calls back to something that's in our brain. So if we look back into our brain's files and we say that RNA polymerase 1 transcribes rRNA genes, then we know that rRNA is a um then we know that rRNA is crucial in making ribosomes. That's another crucial part of what you need to pull out of your brain. That RNA is linked to ribosomes. RNA polymerase 1 is linked to rRNA, which is linked to ribosomes. That's our first linkage. And if you knew that, you would know if it folds incorrectly, then ribosomes would no longer be produced, is looking like a pretty good answer. Now, we can also, if you don't happen to know that information, we can also try process of elimination. So, first of all, as I've just hammered into you, protein's function is heavily dependent on its structure. And so B, where the function is independent of its structure is completely wrong. This is super wrong. Protein structure and function, they are inseparable. They are conjoined twins. You cannot separate them. We can also look at there's tRNA would be processed incorrectly. Really, RNA polymerase has nothing to do with the processing of tRNA. I mean, there's RNA polymerase, believe it is RNA polymerase 3. Yes, it is RNA polymerase 3 that transcribes DNA, but that really has nothing to do with the processing, so you could probably cross that one off. It would honestly, that one comes down to whether you know that RNA polymerase 2 um, codes for the genes that create proteins. But at this point, you've got a 50-50 shot, so that's a good way for eliminating. But basically, what you need to know is there's usually one crucial piece of knowledge. There's also another piece of knowledge. Fully incorrectly changing its structure. That probably means it's not going to work, which means that ribosomes would no longer be produced because they don't have that essential piece of rRNA that is part of the ribosome. So really, if I was taking this test, I had no idea what RNA polymerase 1 did. Specifically, I just knew that it helped synthesize RNA. I would probably be able to cross out these two, and then I would pick between these two. And that's when I think you should put a marker down and say, come back later, because often there might be a question that actually gives you a hint as to which one of these choices it actually is. Okay, so I hope that video is really helpful for you. Please let me know either if you're my student, email me, or in the comments of this video. If you have any questions, I will do my best to answer them, and what you would like to see for the next video that I make for the next unit. And good luck studying. Have a great rest of your day.